Okay, so we're going to move on to the second presentation from Florian Audet on conduct of reproducibility experiment in clinical research. Thanks.
Thank you very much. So let's go. Okay, so as I told you, I had quite little time to prepare the slides. So sorry if there are some slides that some of you have already seen. For instance, this one. This one is a citation by Richard Orton. The case against science is straightforward. So much of the scientific literature, perhaps half, maybe simply untrue. So I gave you the citation just before. I give it to you now because I want to stress the point that he knows what he's talking about. For instance, there is this research paper that is super important because we had this pandemic that was problematic. And this is a phase 1-2 study of the Sputnik vaccine, so one of the vaccine for COVID. And if you have a close look at the figure, you will see that there are some problematic patterns. So those are, level of antibodies at day 0. So at day 0, nobody has antibodies because it was the selection criteria, not have been in contact with COVID. At day 14, you have some antibodies that are appearing. And at day 21, you have antibodies that are here. And that are here, and they stay here. This is a problem. Because there is an excess of homogeneity in this data. Because the data here, the distribution, looks exactly the same as the distribution here. The distribution here looks very similar to this one. This one is pretty similar. There is just one point that is here or here. So there is an excess of homogeneity, which was stressed by some authors like Enrico Bucci and his group. And they asked for a detailed answer. And actually, Nature, again, made a story about this and said that some researchers questioned the Russian COVID vaccine. And they said, the Lancet declined to comment, but said that he has invited the author to respond and that they will follow the situation very closely. So actually, they followed the situation very closely because they published the phase 3 trial in the same journal. And actually, in the phase 3 trial, you can see curves that we often see in clinical research. So Kaplan-Meier cumulative incidence curves. And if you have a look at that, you can see that you have people here that are gained to follow up. So usually we have people that are lost to follow up, but this time we have people that are gained to follow up. So actually the authors will say that this is a typo. But then you can look at this analysis, and this analysis is super important because it gives the efficacy of the vaccine overall and by age group. And you know that the activity of the vaccine by age group was very important because we wanted to have a vaccine that was efficacy for the older person because they were the most vulnerable at the first stage of the pandemic and during the pandemic. So, and you can see that the efficacy is 91.6 overall, exactly like Pfizer and the others. But you can see that by age group, the efficacy is very homogeneous too, despite the fact that the confidence intervals are quite wide. So actually, we should expect, just by random fluctuations, values that are between, let's say, 70% and 96%. And actually, this is pretty homogeneous. And there are also other problems of homogeneity. So it was long to get this published in the Lancet. Actually, we found also problem of excess of homogeneity between the interim analysis of the trial, and the Lancet refused to publish that because they said that they are taking care of what is published in the journal, and the interim results that were published in press release are not of their business. Okay, and this is what we could publish. And if I discuss this example, it's because it will be very important to have the data, to look at the data, to look at the pattern of the data we can have. And if you ask to the data, so for the first phase 1, phase 2 trial study, it was said that you can ask for the data. So we asked and we never received an answer. And for this one, we didn't ask because it was written in the paper that if you ask the data, it will go to the security department. So actually, we decided not to ask the data. But you will say, it doesn't work, sorry. But this is just to say, because you can say, okay, this is a security department of Russia, and now there is this war now, and so on and so on. But actually, for all other vaccines, so I don't know why it's like that. I hope that it's the only slide that is with black and white. But individual patient data were not available on due time for many of the vaccines. So data sharing was not so important. And if I talk about that as an introduction thing, is because I will give a lot of examples around clinical trial data sharing in this. So I was thinking that the title actually was not so easy. But data sharing allows for several quality controls, and actually we have several examples of reanalysis of trials. And for me, as a psychiatrist, I know this example that is a study 329, a study about the efficacy and harms of paroxetine, imipramine in treatment of major depression in the youngest, in the adolescent. And actually, it was published in the original version as positive and has been well tolerated, while it was not the case. And actually, those kind of reanalysis that we would have loved to do with the Sputnik vaccine, that was done, I would say, 10 years after for the study 329. Actually, this is pretty rare in the literature. If you look at reanalysis of randomized control trial, it's difficult. It's difficult to find some. This was in 2014 when they tried to look at all reanalysis. So it was a systematic review of all existing reanalysis. And they identified, let's say, 37 reanalysis. Among those, only 5 were performed by an independent group. So among the whole body of clinical trials we have, reanalysis of clinical trials was almost non-existent. So actually, I was working on this slide yesterday, and I found, wow, reproducibility experiments, meaning when we talk about experiment, we talk about something that is like interventional research, and interventional research on reproducibility in the field of clinical trial. I say, how I can find an example of that? It will be super difficult. So I say, I will try to see how I can make the title. So first, I would say something very important. So I will talk rather about experiment, but about meta-research studies. And I will not solely speak about reproducibility, but I will speak also about transparency. And I think that when researchers are interested in studying reproducibility, very often they make they omit to make a difference between transparency and reproducibility, and sometimes there is confusion between both concepts. So that's why I put that. So I will certainly make some confusion, but it's now in the title, so I can do it. But for data sharing and clinical trial data sharing, now we will have some policy. So up to 2014, there was almost nothing. But in 2016, the ECNG said that it is an ethical obligation. So it's super important, an ethical obligation. And they encourage feedback on a proposal to make clinical trial data sharing mandatory. We can tell that there was a little bit of pushback. And actually, 1 year later, they came with a proposal that is now not solely a proposal, it's a requirement, but their requirement is just to include a data sharing statement in the research paper. It can be yes, I will share the data, or it can be no. And it also implies to declare your intention to share your data in registration. And this is quite important because if you are interested in clinical trial data sharing, you have to plan this very early in the process in order to be able to inform participants that data will be reused, especially for us in Europe because of the GDPR and so on. So this is actually super important. But it's not mandatory to share data for the ECMG. But we can look at what journals have done before making this mandatory, making this a requirement to write a data sharing statement. And we have some examples. So for instance, Annals of Internal Medicine was the first journal to encourage clinical trial data sharing since 2007. there was the first one, and they encouraged and they did not make it mandatory. And when you discuss with folks at Annals of Internal Medicine, some say that they didn't wanted to make it mandatory because they didn't want to lose the randomized control trial, because they fear that people might go away to other journals with the trials if they are obliged to share the data. The BMJ led also the thing because it encouraged data sharing in 2019. In 2013, it made it mandatory for drug and devices. And in 2015, it made it mandatory for all clinical trials. And in 2023, it made it mandatory for all kinds of research, including observational research. So I have now a new one here, but okay. And the others followed the ICMGE at this point. When I collected the data to make this figure, I remember that I contacted the editor of BMC Medicine to ask them if they have a data sharing policy, and actually he answered that he didn't know. But we can look at what happened for the first experience. So for instance, we looked at BMG and PLOS Medicine, and we tried to get the data. And we tried to get the data under a mandatory policy in order to reproduce the analysis. To us, it was important to have both the question of transparency, can we get the data, and the question of reproducibility in this study, because often people won't share you the data just to show that it's feasible. You know, it takes time, it takes cost for them. So if they share the data and you don't do nothing with the data, it can be quite difficult. So I'm talking here about clinical trial data, which is quite sensitive. So we said it's for the purpose of reproducing the analysis. And actually, we had a registration for that. So we were able to show people what we wanted to do with their data. And actually, we got the data for almost half of the study. And some will say, no, this is very bad because they all promised to share the data. But actually, it's very good because we were at a level of 0. And some didn't want to share the data. They said, OK, I would be more willing to share it for, let's say, individual patient data meta-analysis and so on. So mixed results, 46%, 50% almost. And we tried to reproduce the analysis for the study where we got the data. And in general, we were able to reach exactly the same conclusion. Although, in a few trials, we found some mistakes. And we reached the same conclusion, but there were mistakes for one in the code. For a second one in the... We don't know where, because when I asked the researcher where was the error from, they agreed that there was a mistake, but they didn't know where it came from, because they didn't use a code, so they use a click button software. And one paper did not provide sufficient data in methods, meaning that reproducibility is not just a matter of having the data to reproduce analysis, but having sufficient information to know what was done. So we can look at what Annals of Internal Medicine has done, and Annals of Internal Medicine encourage, since 2007, clinical trial data sharing. So we looked at all the studies as published. So we found several randomized control trials published in their journal. And for all these randomized control trials, we searched at all citations. So we found all citations of those randomized trials. And in each and every citation, we looked if it could be a reuse. So a secondary analysis, an individual patient data meta-analysis, or a reanalysis. And even meta-analysis aggregated data, but this is more anecdotal. And we found no reanalysis, of course, but we found a few secondary analysis and pooled analysis. So we try to look at individual RCTs published in the journal if intention to share data changed over the time period, over the 10 years of the policy. And actually, it improved a little bit at the beginning. Then no constant increase. We could have hoped that it would have increased across time, but it did not. But we had a group of journal sharing the data and a group of paper sharing data and a group of paper not sharing data. We could compare those groups, and we found no difference. There was no gain of follow-up in this study, which is reassuring, and it's good. And we can look also at what happened when the ECMG adopted the policy. So for instance, here in 2018, so we looked actually in the ICMG journal members. There were 14 at this time. And we look at affiliated journals, so journals that claim that they follow the ICMG. And among the ICMG journal, we found that they implemented correctly data sharing statement with nearly 100% of data sharing statement in the journal, with expression to share data, intention to share data in 77%. It's not exactly the same for all journals that claim to follow the ICMG. Journals were very heterogeneous in terms of data sharing policies. Being part of a given publisher group was very associated with adoption of a data sharing policy, meaning that it's not directly the journal, but rather the publisher. And also, we found that in those randomized control trial published in those journals, data sharing statements were quite rare, and intention to share were found also very rarely. So we could look also at pre-post experience to see if we can get the data and reproduce the analysis. So we did this in a surgical journal because it was with a colleague who is surgeon. He was interested in that. And we look at 65 studies before the ECMG policy and 65 studies after the ECMG policy. So actually, the ECMG policy changed something. There was no data sharing statement. After the policy, we had 11, so not that much. And actually, among those who intended to share the data, none shared the data. We were two here. We got 2 datasets before the policy, 2 datasets after the policy. So you can compute a p-value in your head. This will be more difficult to do in your head because, but we look at the 4 studies we had, and actually we had quite similar results when we try to reproduce the results. And here it will depend also on what you call reproducing a result, because for instance, in one of the studies, we got exactly the same numerical estimate than in the paper, but we figured out that there were 50% of missing data in the study. This was not much mentioned in the paper. So we got the same result, but we got a few more details that are quite interesting. We tried to do that by reanalyzing studies. So it was actually a registered report, this one. We tried to do that by reanalyzing studies used for pivotal trials used by the European Medicine Agency. And we selected, again, 62 studies. So 49 were from companies that have a data sharing statement, and 13 were from companies without a data sharing statement. And we look and we ask the data to reanalyze the study. And actually, we were able to reproduce the analysis for only 10 studies. For 52, it was not possible despite they had a data sharing statement. So they had a data sharing policy. They had always a good reason, and one of the most frequent reasons was that the study that was used for the approval of the drug was still ongoing. For instance, it was still ongoing for several safety outcomes that were being taken after. But we wanted just the primary outcome, the one that was used for the assessment by the EMA. And we got similar plots to reproduce the result. And actually, results were pretty well reproduced, except we found one analysis that would not have been done according to the protocol. And of course, that was done to make the study positive. But in general, we got effect sizes that were quite in the same range as effect sizes that were in the publication. So actually, we can be very positive when we get the data in general. We get the same result. And actually, this is a good result and a strong result. I feel even more confident when I'm able to reproduce the result of a given study. But from what I discussed, I found that there is a bunch of limitations. And limitation is what are we looking at? We are looking at a lot intention to share data. So if people want to share data. We are looking a lot of sometimes if people actually share the data. But in the literature, you find very low information about how much of this data is reused when it is shared. How much of this data is reused and give rise to something like a publication or conference and so on that, and how much impact this does. Because data sharing is like a pipeline, and the outcomes we are looking are very early in the data sharing pipeline. And actually, I'm not very interested to know that the people of the Sputnik trial are willing to share the data. I'm more interested if I can reuse the data, see if there's something wrong or if I can be confident or not, and see if this can have an impact. For instance, for the COVID vaccine, the Sputnik was used a lot in South America. So actually, having accurate information on that would have made an impact. And we looked also at the type of studies, and you see experimental studies, there are very few experimental studies on clinical trial data sharing. That's why the title was not so good. And that's why we need to think about, in reproducibility experiments, outcomes and interventions that are aligned with the three ideas that I told you just before. I will give you an example, because I had the idea to change the title to Thank You Coffee Break, because there is nothing to say about reproducibility experiment. And if you got the coffee break, I will recommend you to take my meringue again. And here is a recipe. And here is a kind of reproducibility experiment that I would love to do with, for instance, clinical trial data sharing or any kind of open science practices to see if it actually improves research reproducibility. But we did this with meringue, and this is exactly the way I used to talk about the public about that. So it's positive. I give the recipe. Everybody understands it. So what we did was, so I blame the recipe. I said, this is not my fault. This is the fault of the recipe. So with Constant, that is here, we decided to make a randomized control trial to explore that. And as I told you, there is an Equator reporting guideline for almost everything. So we searched the one for meringue, and we found one that is quite useful. It's the tidier checklist that helps you to give sufficient data for a given intervention to make it more reproducible. So we planned everything before editing the recipe with tidier, but we planned to see a baker and to ask him to help us to improve the recipe with much more detail. So we followed tidier, and we followed tidier. So I see some people who are interested, but let's wait for the results. And we tried to frame the research question like that. So with the concept of research reproducibility in mind, improving the recipe is basically improving methods reproducibility. But what we want to prove is that by improving method reproducibility, you improve research result reproducibility, and you also improve inferential reproducibility. So then when you have that, you have to build the outcomes that are able to capture that. So for example, result reproducibility, we decided that it looks nice. You look at the picture of the original recipe, and you compare the meringue that you get to this. It should be also tasty, I hope, because in the recipe it said that it's super tasty. And in order to explore the idea of conclusion, we found that it could be good to organize a big sale of those meringues and to see if people decide to buy a meringue blindly, independently of the group where the meringue comes from. And it was good because we could speak about that with students, because they were fun, so it was quite fun to do with them. So for those who agreed to participate, and it was good because we were also able to talk about that with the public. So here is a baker that helped us. He does very nice meringue. So actually, in order to improve method reproducibility, we also made a little video, because sometimes with a video you can show things that you cannot write very easily. For instance. There is some very technical stuff with meringue, like the bec d'oiseau. I don't know how we call this in English. And then this is the result. So we gave everybody a standardized box with eggs and all the things to cook. So here are the box. And then we ask people to make the meringue, and we put those on a table, and people could come and eat the meringue. And actually, we sold the meringue. We even were able to assess the survival of meringue. And I must say that, no, this is very different from medical experiment, because survival of meringue is good when it's short. For medicine, it's the opposite. And then we had the jury here who rated all the meringue on our outcomes. And this was so it was super fun. We discussed with a lot of people. And this is a forest plot of Christmas trees. So, so as you can see, this is the primary outcome. And actually, it's negative. So it means that it's not the recipe, but the baker. So I have to admit this. And we have to admit our failure. So does it look nice? No. Does it taste? No. Does it improve reproducibility? Actually, no. So nothing. So this is a negative trial, and this is super important because we could have tried to torture the data or so on, but we did not, because meringue deserve much more than this. And okay, but I need to talk about clinical research just a little bit, and I think I won't have so much time, but I will just be fast on that. Just to say a few things. So people are able to publish pyramids in a BMG, EBM. So pyramid, you know, all know this pyramid of, you know, what is trustworthy, what is valid in science. So case theory, case control study, cohort study, randomized control trial, systematic review, meta-analysis. I bet that this pyramid was drawn by somebody involved in the field of systematic review and meta-analysis. But this is a pyramid, but we were able to publish a cube. So this is our cube. Much more fun. And we did this with Johanna, Nicolas, and others, Sabine also. And we decided that we can agree with everything with the reviewer, except that we want to keep the cubes. And actually, we can envision medical research with this kind of idealist and very simplified thing, but that we can think about flexibility in terms of analysis and so on. And actually, we have method flexibility that can be fixed, especially in interventional research, and not so much in observational research. We have now another degree of feasibility due to measurement, because we have a lot of routinely collected data, in contrast with data collected for research purpose, where you decide exactly what you want to collect, how you want to collect this. And we have now another dimension included by the fact that we are going to share the data, raise data by user, that is due to flexibility of research hypotheses between primary analysis and secondary analysis. And what is true is that, for instance, registration only concerns this part in interventional studies. So this morning we say, no, it's a mess in a prognosis model and so on, and it's not a mess in clinical trial. We said why? Perhaps this is because we have had a lot of scandals with drugs and a lot of deaths, so we try to make things a little bit more, or less, I would say, less flexible and easier to control. But I think that the problems are mostly everywhere at different level and different degree, and especially they can be here in secondary analysis of data. So this was just to talk a little bit about clinical research, because I don't have much more time, and I would say that I had a lot of many other slides, but that this one is perhaps one important one, because what we did with meringue is something we want to do with researchers. So instead of randomizing cooks, we want to randomize research project. And I think that we really need a coffee break. No, no, but we really need actually to have interventional research on reproducibility, and especially in clinical research. And we need to have experiments and not solely studies, observational study, because as we said since the beginning, meta-research is also concerned by all these degrees of freedom that we discuss here and here. Thank you very much.
Florian, for a great presentation. Some questions? Perhaps as you highlighted the flexibility of the analyses and the registration of randomized controlled trials, what's your view on the registration of observational studies?
So my view that it would, so it's the title, it will improve the transparency and reliability of observational studies to register those studies. I think there are some pushback for that from the field of observational research, but actually we have many responses to those pushback. One pushback could be that people fear to be scooped because they will declare what they want to do. But actually, with the Open Science Framework, they can put an embargo. One other pushback would be that it will force people to be dishonest because they will have to perform their data first, register it, because it's sometimes retrospective, and because they will be forced to be dishonest, they will be dishonest. I don't think so. I'm very positive, and I think that people are dishonest. They really exist, but they don't need to be forced. So they are dishonest by nature, so it's not a problem. And for many of the researchers, you want for them to be dishonest. Another pushback would be that with registration, you cannot do exploratory research. No, it's not. It's not the case. You still be able to do exploratory research, but you will be able to label it as exploratory research. This is quite different. So actually, I think it's as needed as other. But we need to be perhaps much more flexible because there are something that you cannot define a priori. So you have always a lot of surprise, and especially when you are in this area, you will have many surprises. But you can specify a priori the rules to decide to handle those surprises. You can also decide a priori that certain areas are not totally decided because we cannot know, and that for those areas there will be some room for degrees of freedom, and it allows you to specify it. So I think it's possible, and it will work, but it will also need a little bit some change because the template used in clinicaltrial.gov is not perfect, so you can use exactly the same template. But if you look at the project that was called Duplicate RCTs, where they duplicated RCTs with observational data, they were super transparent, and they put their protocol as an additional material to the registration on clinicaltrial.gov, and it worked actually.
Yeah, thank you very much for this inspiring and also very funny talk. My question is, or maybe a suggestion actually, that suddenly occurred to me is after your talk, you talked about methods for reproducibility, and reproducibility is not sexy in many fields, particularly in the RCT field. There's a drug developed, manufactured by a company. There's typically one big trial done. They get FDA or EMA approval, and that's it. Nobody says, hey, let's do another trial on that drug. And the same with diagnostic tests. A test is manufactured. They go to the MDR, IVDR. They do an accuracy study, if they do it anyhow, and then they get to the market, and that's it. Maybe usually the prediction model. I'm trying to make links between talks also. The prediction modeling world learns everything about trials and DTA studies. We are lacking. They are lacking behind. But maybe here it's the opposite. For the very first time, I realized that, because if there's one thing that is in the DNA of prediction modeling, at least in the guidance, is that once you develop the model, you need to do multiple validation studies before applying it to practice or in your patients or in your clients or whatever the stakeholder group is. So having said that, is reproducibility is in the DNA of the guidance of in the prediction modeling world. So perhaps for the very first time that the trial world and the DTA world can learn a little bit from the guidance that that comes from that prediction modeling world. Because if we don't support and keep on telling the message, let's reproduce what others have done, because that's really valuable, that's how we change practice, then I think then we are making steps forward. So keep on repeating this message, and you might borrow here from that other field. But maybe you have another opinion. Can you?
No, thank you. I fully agree with you. And actually, it reminds me also a little bit, but it's perhaps not directly in line with that, but with the approach of statistical significance by Fisher and the one by Neyman-Iperson, that is perhaps not exactly the same. That is an old story for us, actually, that ended in something that is in between both, but is still not totally understood. So I find it enlightening.
Yeah, it is actually strange that the FDA and also the EMA just desire one big trial from the company itself usually, and that's about it. And then they, approve it or not, of course, depending on the result. Actually, it's interesting that the world is going for one big study. What's one study? If you apply it then to the whole world,
So
just an idea.
For my field, so I had so in my field, I would say in psychiatry, we generally have generally more than one pivotal trial. It could be 2 or 3, but there is a tension now to decrease the bar with accelerated approval and so on. So in my field, psychiatry, we got less and less things that are reproduced. So actually, I would say that also we are losing something in the field. We are not getting inspired by you, but we are losing things.
It's a very nice talk, Florian. As usual, my feeling is that replication of the analysis of randomized control trials is probably easier to do just at the time the first analysis is done. Yeah. And it must be, in my view, it could be something that journals ask for. Some people say that these data are too precious to be analyzed only by one team. Yeah. And my view is that it's easier to do that at this time rather than to do that 3 years later or 10 years later. Yeah. And it's the same for observational data, in fact. Yeah. And in addition, for observational data, it's probably easier to replicate in another database or
I agree. So I would say that for statistical analysis, the FDA is pretty good because they have their own statisticians that are looking at the data and reanalyzing the data. And sometimes there are mismatches between the publication and what the... Yeah, exactly. Yeah. And I fully agree also that it's much more easier to replicate things from observational studies in another database. As for instance, the example of suicide that I give you, suicide and full moon, they were able to reproduce it in 2 different data sets. And I think it's part also of the culture in epidemiology to have replication before getting a strong feeling that there is a signal or something.
Okay, if there's no more questions, thank you very much, Florian.